Next category of microorganisms is protozoa. So protozoa are classified under Protista kingdom according to Whitaker's 5 kingdom classification system. The principal characteristic of protozoa is that they lack cell wall. So this characteristic is similar to the characteristic of animal cells. So that is how they resemble with animal cells. The diameter of protozoans is about between 5 to 250 micrometers, and the study of protozoa, it is called as protozoology. Protozoa occur in different habitats, including seawater, soil, and fresh water. Protozoa can be parasitic, that means they are dependent on other organisms for the growth, and in that process, they may cause harm to the host cells. They are called as parasitic protozoa. It is a kind of negative association that occurs in animal groups. So these parasitic protozoa are responsible for the production of several diseases in humans and animals that we'll see in next few slides. They are divided into two categories, basically free living protozoa and symbiotic protozoa. Symbiotic means the association. So this association, it can be positive association or negative association. The optimum temperature required for their growth is from 16 to 25 degrees Celsius, and the maximum is from 36 to 40 degrees Celsius. It is very interesting to note that the few species are found in hot water springs at temperature of as high as 56 degrees Celsius too. And these protozoa, they exist in the form of resistant form, which is called as cyst. Symbiotic protozoa occur in different associations like commensalism and mutualism, as well as parasitism. Parasitism, as it is mentioned, that it is a negative type of association which are responsible for the cause of different diseases. Morphology of protozoa. So these are eukaryotic protists. They are unicellular and they have different shapes and sizes like spherical shape, oval shape, slipper shape, or they may be pleomorphic as seen in case of amoeba. The intracellular structures are contractile vacuoles, which are necessary for osmoregulation, the food vacuoles, which is necessary for storage. So rest of the intracellular components are just same as like of the eukaryotic cell. The nucleus. Unique characteristic of protozoa is that it may have more than one nucleus. So many of the protozoa, they have one nucleus only, but few of them have some extra chromosomal DNA. That is, they are having multiple nuclei, that is from 2 to 8. That type of nucleus, it is called as micronucleus, whereas chromosomal is called as macronucleus. So in several species, each individual organism may have two similar or dissimilar nuclei. When the two dissimilar nuclei are present, one is always large macronucleus and the another one is micronucleus. The nucleus is surrounded by nuclear membrane, as it is a characteristic of eukaryotic cell. Micronucleus, macronucleus controls the metabolic activities and regeneration process. So major activities of a protozoan cell are controlled by macronucleus, the DNA which is present in macronucleus, whereas micronucleus, it is important role in sexual reproduction. The component of protozoan, that is the covering, it does not contain cell wall, so it contains only cell membrane. So the coverings which protect the cytoplasmic contents of a protozoan cell are plasma lemma, and there may be other types of covering. So cytoplasm, it is separated from the external environment by cell unit membrane, and that membrane is called as plasma membrane, which is quite tough to resist the external environmental conditions. So it provides the protection but also controls the exchange of substances. Sometimes this membrane, it may be called as a pellicle, a differentiating tissue. So it is important role, it is important in the pinocytosis process, that is the uptake of fluids or the soluble nutrients, or it is also important in relation to the binding of cell to the solid substratums. Some protozoa have coverings over this pellicle, so that is the extra protection, and it is called as theca or shells, tests or lorici. These coverings are generally uh, formed because of the accumulation of calcium carbonate and silica from soil and water. The feeding structures present in protozoa, say for example, amoeba. Amoeba gather food by means of a pseudopodial engulfment. So it entraps and engulfs the food particles with the help of an extension of the cell membrane called as pseudopodia. Whereas in case of ciliates, the cytosome is the actual opening through which the food is ingested. That means this ciliates like paramecium, it actually has a mouth like part, which is responsible for the ingestion of food material. Another characteristic of protozoa is the formation of cyst. Many of the protozoans, they survive in dry conditions by the formation of resistant cysts or the dormant stage. These cysts can survive adverse environmental conditions such as desiccation, low energy supply, or under anaerobiosis conditions. So extreme environmental conditions, these protozoa, the vegetative cells of protozoa get transformed in the form of cysts, and these cysts are resistant to unfavorable environmental conditions, and this is how a protozoa can remain viable under these abnormal environmental conditions. The locomotory organs in protozoa are very important because that is the basis for classification of protozoa. Locomotory organs means the organs which are responsible for the movement of protozoa or the motility of protozoa. These organelles are pseudopodia, flagella, and cilia. So in addition, a few protozoa without such organelles can carry out a movement on the solid substrate by the body flexion. These movements are called as gliding movements. A pseudopodium, it is a temporary projection of the part of cytoplasm. The flagellum, it is extremely fine filamentous extension of the cell. The number of flagella on the protozoan body, it ranges from 1 to 8. And the third type of locomotory organ is cilia, which are the small projections present outside the cell membrane. Method of reproduction in protozoa. So protozoa can reproduce by asexual reproduction and rarely by sexual reproduction. Asexual reproduction involves binary fission, budding, or multiple fission. In case of binary fission, say for example in this diagram, the binary fission of amoeba cell is shown. So this shows that the division is longitudinal. One cell divides into two equal cells. Second one is budding. This phenomenon is same as we have studied in case of yeast. There is a protozoan cell, a small bud protrudes out, and then that bud, it grows and ultimately it leads to the formation of a separate cell, and it separates from the mother cell. Third type of division is multiple fission. So as we can see in this diagram, which is showing multiple fission. So extreme left is a parent cell, that is a protozoan cell, which contains a single nucleus. So whenever there is this division occurs, so this nucleus, it divides multiple times. It divides multiple times by the method of mitosis, as we have seen, and it leads to the formation of several nuclei. These nuclei, they get further compartmented by means of the cell membrane. So separate cell membrane is formed, that leads to the formation of daughter cells. These daughter cells are called as cysts. These cysts, which are in more than 2 number, these cysts then released out of the cell as a result of the lysis of the cell membrane of parent cell. And further, this cyst, when it gets favorable conditions for the growth, it gets matured, and the mature form is called as daughter cell. So this is how the multiple fission occurs. Then, in case of sexual reproduction, so sexual reproduction involves fusion of two different sex cells or gametes. Means these are nothing but the protozoans having different gametes, which results in the formation of zygote, and further this zygote then develops. So this is called as syngamy. Another way of sexual reproduction is conjugation. So what happens in conjugation, that two cells having genetic material, that is a micronucleus, several sets of micronuclei, these two cells, they combine together, and then there is exchange of genetic material between these two cells, and then after that exchange, means the genetic material from one protozoan, it gets transferred into another protozoan. In the same way, the genetic material from another protozoan is transferred into the uh, first protozoan. So this is how there is exchange of the genetic material, which is called as conjugation. The classification of protozoa, we'll say it a little later. Yeah, so this slide shows the process of a combined process of conjugation and syngamy. So the process of conjugation, it is a cycle, so let us begin it from here. So we can see that there is presence of a macronucleus at the top. This is the process in case of paramecium. So there is a macronucleus at the top in both the cells of paramecium, and we can see the micronucleus are shown in colored, blue colored and red color. So these are the haploid micronucleus. Haploid means the stage is N. Then in the second step, few of the haploid nuclei, say for example here it is shown in a green, blue and red, there is exchange of this micronucleus. So red one is going towards in the opposite protozoan, and the blue one is going to another protozoan. So there is exchange of protozoans. So after this stage, these two cells, they may get separated. This is called as conjugation. Sometimes there may be further development, which is called as syngamy. So what happens in syngamy is that these two protozoans, they fuse with each other, and as a result of fusion it leads to formation of a single cell. Now the single cell is having diploid nucleus, so it is having two sets of nuclei, and that is why it is shown in green color. So it is a diploid micronucleus. So this diploid micronucleus then further divides, and whenever there is binary fission, each paramecium will receive a set of nucleus like this. So this protozoan then divides, it leads to the formation of daughter cells now which are having diploid nuclei, and this is how the process continues. Let's go back to the syngamy process. It is a fusion of two gametes in sexual reproduction in protozoans. It is of three types. Isogamy. Isogamy means two protozoans, means two similar gametes, means structurally they are similar but genetically they are different, so they are marked as positive and negative. So when they fuse together to form a zygote, the process is called as isogamy. So this isogamy process is common in the protozoan called as monocystis. Second type of syngamy is anisogamy. Anisogamy means opposite to isogamy. Here we can see that the gametes are dissimilar. So structurally they are dissimilar. So these two gametes then combine with each other to form a zygote. This happens in case of Plasmodium, a malarial parasite. Then third type of syngamy is hologamy. In hologamy, we can see that these protozoa are mature protozoa, and these mature protozoa they combine with each other to form a zygote. This is commonly seen in case of Trichonympha. It is the name of protozoan. So these are the three types of syngamy. Remember that syngamy is a sexual way of reproduction in which two opposite mating types they combine with each other to form a diploid cell or a zygote. This is an example of hologamy. You can see the protozoan which is having two separate cells, then they in the B step they are getting combined, and in the C step there is formation of this zygote. Then the zygote may further develop into individual protozoan. These are the few examples: paramecium, amoeba, trypanosoma. This is the picture of paramecium. We can see at the center there is a nucleus. Outside of the cell membrane you can see very fine bright colored cilia. They are multiple in number. Pleomorphic shape means there is no definite shape. It is a characteristic of amoeba. You can see at the center there is a nucleus and several intracellular organelles, granules, storage particles present in the cytoplasm. We can see the extensions of the cell membrane which are called as pseudopodia. This is the picture of a Plasmodium, which is a malarial parasite. In the life cycle of this malarial parasite or Plasmodium, there are multiple steps and multiple shapes. This is one stage which is called as trophozoid stage of a Plasmodium. This is Trypanosoma, which is having a flagellum, and it is responsible for, so it is motile by flagellum. This is this is plasmodium, which are non-motile. This is amoeba, which is motile by means of pseudopodia, and this is paramecium, which is motile by means of cilia. So there are four categories of protozoa. One is which are motile with cilia, second one which are motile with pseudopodia, third one non-motile, and fourth one motile by flagella. So this leads to the classification of protozoa. So we'll go back to the general characteristics again. Okay, this is the classification scheme. So classification is based on presence or absence of locomotory organ, and if locomotory organ is present, what type of locomotory organ is present. If it is flagella, then that category is called as flagellates. Subphylum is Mastigophora. So these protozoans have one or more flagellum. Examples are Trichonympha campanula, Giardia intestinalis or Giardia lamblia, Trichomonas hominis, Trichomonas vaginalis, Trypanosoma gambius, and Trypanosoma rhodesians. These are the examples of flagellates. Second class of protozoa is amoebas. The subphylum is Sarcodina. These are motile by means of pseudopodia. The example is entamoeba. Third class is sporozoa. Phylum is Apicomplexa. The protozoans which are classified under this class are non-motile, so they do not contain any organ for motility. The example is Plasmodium. Then the fourth class is ciliates. Phylum Ciliophora. So as the name indicates, the protozoa which are classified under this class are motile by means of cilia. The example of this class is Paramecium. So these are the four classes of protozoa. We have seen this classification and sexual and asexual mode of reproduction in protozoa, the syngamy. Then we'll go to the final part, that is importance of protozoa. So protozoa are environmentally important as well as clinically important. That means they exist in several associations like parasitism, which is a negative association. They also exist in mutualistic type of association, which is a positive type of association, and there may be several other important aspects of protozoa. What are these? Let us say human and animal pathogens. So there are few protozoans which are responsible for production of diseases in humans and animals. Say for example, Giardia intestinalis. It is associated with diarrhea in children and infrequently in adults. Second, Trypanosoma hominis, that causes diarrhea. Trypanosoma vaginalis, it causes vaginitis, that is the inflammation of vagina. So these are the infections caused by trypanosoma. Then Entamoeba histolytica, which is a type of amoeba, it inhabits the intestinal tract of vertebrates, and it is a major cause for amoebic dysentery in humans. There are two types of dysentery that can be seen in case of humans: bacillary dysentery and amoebic dysentery. Bacillary dysentery is caused by bacilli. Bacilli means bacteria, and amoebic dysentery is caused by amoeba. That is the difference that you need to remember. Toxoplasma gondii is the causative agent of toxoplasmosis. So the symptoms are just similar to meningitis and hepatitis, and it is one of the very dreaded and life-threatening diseases. Now, other examples, important examples are the pathogens, human pathogens, Trypanosoma gambians and Trypanosoma rhodesians. These two have a clinical significance, and these two protozoans are flagellates. They are flagellates. They are responsible for a life-threatening disease, which is called as African sleeping sickness or African trypanosomiasis. So as the name indicates, it occurs in the region of Africa only. That is due to the presence of this TT fly. Sometimes it is called as tsetse fly. So this is a vector. This fly is a vector which carries this pathogen, which is born from the blood of infected individual, and then so that is through bite, and then it is transferred to the other healthy individual, and this is how the transmission occurs through this tsetse or TT fly. So this is the picture of this TT fly having a bite on human skin. So from the infected individual, the organisms will be sucked by means of blood, and when this infected blood which is transferred to healthy individual through another bite, the transmission of this pathogen it may occur. This is the pathogen, that is trypanosoma. You can see this is, and the rest of the things are blood cells. The circular one are blood cells, and the other white ones are white blood cells. Red one are red blood cells. Malaria is another important disease, a very common in throughout the world, which is caused by Plasmodium. So there are several species of Plasmodium like Plasmodium vivax, Plasmodium falciparum are more common, are responsible for the production of disease through the bite of infected mosquitoes. That is, female Anopheles mosquito is responsible for the transmission the same way in which TT fly does. These are the different symptoms that occur due to uh, malarial parasite: headache, fever, fatigue, pain, chills, that means shivering, sweating, dry cough, enlargement of spleen, and nausea and vomiting. So the critical symptom is very high fever. Apart from these disease causing protozoa, there are certain protozoans which are beneficial or which are living in mutualistic association. Now, what is mutualism? Mutualism means that is a type of symbiosis in which both the partners get benefited. Means the cells or the organisms which are in association. So each of the partner gets some benefit because of this association. So mutualistic association is seen in case of a protozoan called as Trichonympha campanula and the termite gut. This Trichonympha campanula, it is associated with the gut of termites, and it is association, mutualistic association. Let us see what is the mechanism. Trichonympha campanula, it has ability to digest cellulose. Now you see termites, they feed on wood. Wood is rich in cellulose, but these termites are not able to digest this cellulose. This digestion is made possible by the intestinal inhabitants, that is Trichonympha campanula are present in the gut or intestine of this termite, and whatever this termite eats, it will go into gut, and this cellulose that enters into gut, it will be digested by this Trichonympha campanula. And in response to that, Trichonympha gets food and shelter, nutrients, protected environment in the gut. And in response to that, termite gets benefited because its cellulose is digested into simple sugars, and this simple sugar is then absorbed in the body fluids and which is utilized as a source of carbon and energy. So these organisms are lying in the termite gut. Termites are unable to digest cellulose, which is a principal component of wood. Trichonympha campanula digests the cellulose by enzymatic action and converts it into soluble carbohydrates easily digested by the insects. So these are the termites, as you might have seen, which are feeding on wood, which is rich in cellulose. And this is the picture of Trichonympha campanula, which is present in the gut of this termite, and they are responsible for the digestion of cellulose, conversion of cellulose into simple sugars.